Back to a series of lectures on quantum biology, and finally today we'll be talking actually about biology, but let me just say a few words leading to it from a physics perspective, and that is sort of one of the common criticisms of the use of quantum concepts outside of physics and chemistry, let's say, is that it applies only to tiny little particles and molecules maybe. But that's not true, because if you know physics well enough, you will understand that there are examples of microscopic, which is any arbitrary size objects which are in a quantum state. And this is kind of a short list of these, the most well-known. Superconductivity, which actually was discovered in, I throw in historical notes, so 1911, if I remember correctly, something like this, or more than 100 years ago. Although in those days, yeah, it wasn't macroscopic, but it was a very low temperature phenomenon, something like until high temperature superconductivity was discovered in 1986. It was limited to something around 10 Kelvin, 11 Kelvin, in the teens, which is what, minus 260 degrees Celsius. So obviously biology doesn't live, at least as far as we know, but biology doesn't apply to such low temperatures. But now, high temperature superconductivity over the past almost 4 decades has cranked up these temperatures. It's a transition from insulator, from metal to superconductor, to not quite room temperature yet, but I think the latest I've seen is something like 196 Kelvin. So we're creeping up to that point. And then superfluidity is another example. So superconductivity is when materials conduct without resistance. Superfluidity is for materials which don't have any viscosity. So basically you create a vortex and it turns around forever and ever, right? So these are macroscopic quantum effects. And then we have well-known laser action. It's also a phenomenon that Einstein contributed to the discovery, and it's definitely room temperature, macroscopic effect. I'm sure you have some laser pointers, scanners, QR codes. Everything involves laser, which means coherent light, so electromagnetic waves at a single frequency. And so that's just like superconductivity, superfluidity. This is a so-called condensation phenomenon, where systems of numerous, in principle, infinite particles are functioning in concert, synchronized completely with frequency or some other property. And the main difference between superconductors, superfluids, and lasers is that lasers require external energy supply. So this pumping of energy creates this excited state from which light is emitted when the electrons come down to the lower energy states. And that, by the way, is very apropos for biology, because as we talked about, and we'll come back, biology requires metabolism, so it has to be in a we call it a metastable energy state. So in terms of energy, I have to explain the physical jargon. We have stable states, which typically mean local minima of the energy function. And then metastable states are also local minima, but not the lowest energy. So you can have several metastable states and one lowest energy state, which is called the ground state in quantum physics. All right. So laser action is, for this reason, very useful to think about, because it in a cartoonish way, is a metaphor for life, because it needs to be, and the energy supply needs to be strong enough or frequent enough to maintain this metastable state. Just like for us, if you are not fed regularly, or if you don't drink enough water, but basically it's about energy. You have to have supply. I don't know how long we can last without food. Maybe 2 weeks maximum, probably. I would say maybe less than 2 weeks. So at any rate, I'll come back to metabolism because this is the crux of the matter, in my opinion, when it comes to quantum biology. And then we have next one, which is also important. Phonons are, and now we're talking about, I mentioned superconductors, superfluids, laser action. These are examples of sort of collective state, collective. That means not a single particle like electron, a single electron or a single neutron, but in principle, in infinity, a whole microscopic, let's say Avogadro's number of molecules or particles are in the same state. Okay, so that's a collective state. And crystal vibrations, so movement of molecules in a crystal, is called phonons. They propagate through the crystal at the speed of sound, basically. The speed of sound depends on the object, changes. Speed of sound in air is different than iron, etc., etc. But there's something very special here, and we'll come back to it. I just want to say that it has to do with the lattice periodicity. So a periodic lattice where you have molecules forming a structure, right? Regular structure, regular occupation of sites. And so these waves need to be confined to the size of the crystal, and they have both the longest possible wave, which is such that the half wavelength is between the two ends of, in that box, crystal, cubic crystal, or whatever it is, and the shortest wavelength, which will be half wavelength, is between the two neighboring molecules. So, and then everything else needs to be quantized such that half wavelength fits between any number of nodes, we call it in a lattice. So positions, right? Molecules sitting there. That's very important. It is really another form of quantization. So we talked about quantization in terms of energies. This is related to it, but quantization specifically in terms of wavelengths fitting into the structure. Waves cannot fit where there's no matter. Okay, so that's basically the explanation. And we'll come back to it because it's an important analogy for metabolic activity of enzymes in the human body or any sort of living system. Semiconductors and magnets I mentioned before. So these are all quantum systems at a macroscopic scale. Some at room temperature, the latter ones. Crystals are kind of interesting because below a certain temperature they are in a quantum state, above they are in a classical state, and there's a sharp, in this case there's a sharp delineation. It's called the Debye temperature, and in some cases you can see the quantum signature, and in some cases it depends on the temperature and depends on the size and the dimensions of the crystal, you will not see it. Okay, so just to summarize quantum physics, we talked about quantum mechanics, and then we mentioned that quantum field theory is the more mature version of quantum physics, where we replace particles by fields, and creation and annihilation fields basically propagate through space and create excited states of matter. We can go into quarks and gluons, but we want to actually zoom out, not zoom in, right? And zooming out to large structures, microscopic, such as biological systems. And as you can see here at the bottom, quantum physics explained everything except gravity. And that's still a conundrum, and Einstein couldn't crack it. Nobody has been able to crack it yet, and whoever cracks this, it will go to Stockholm for free. That's guaranteed. Okay, now, so this is a list of properties the quantum systems offer for us to explore in the context of biology. Why would this be important? Well, first of all, I mentioned before stability of electrons and atoms. Without quantum physics, we wouldn't have atomic structure, because electrons collapse on the nucleus and basically reduce to a point. And now we have sizes and structures, so it leads to complexity emerging out of symmetry. complex molecules emerge out of it, all kinds of atoms, and then you can create this amazing Lego, which is called matter. And the connections between the Lego pieces, bonds, chemical bonds, are quantum. So everything that we build in structure and matter is based on quantum interactions. Sometimes we forget it, but I want you to remember. And then we have entanglement. Entanglement is this weird property that quantum systems, specifically constructed, can be connected across infinite space even. So if you disturb one, the other will feel it or react to it. And that was disputed for the longest time, and Einstein didn't believe in it, and now in 2022 Nobel Prize was given for the experimental demonstration of the reality of this quantum entanglement. And maybe I mentioned this in one of the lectures, but okay. So entanglement, and I challenged you, maybe I challenged you about something else, but entanglement is also a very intriguing property that one could apply to the human mind, to entanglement between human beings, emotional entanglement, or some other kind of entanglement sometimes. Some of you may feel the presence of somebody close to you, even though that person may be at the other side of the Atlantic or somewhere else. Have you experienced any such emotional entanglement with somebody? Felt that you're connected?
Yeah.
Yes? Anybody? Who hasn't? It could be actual sometimes in dreams. And you know, I'm a scientist, I'm not a mystic. In private I may be a mystic, but in public I'm not. But so from the point of view of science, I think dreams, or the dreaming world, the world of dreams, is special because at that point I think we are disconnected from the noise of everyday life, from the chatter, and therefore probably through dreams we may have a better sense of connectedness with, you know, this entanglement with others. So one last time, is anybody here who has not felt connection, either in dreams or awake? You haven't? Okay, wait, just wait.
It'll happen.
It'll happen. Okay. All right, so we have entanglement, coherence. I told you coherence is really synchronization. And that coherence is also at least a metaphor, if not something real, for living systems of this unified sense of being, in spite of the complexity, the diversity of cells and tissues and molecules inside. Everything seems to be so messy, yet it's organized and functions as one, coherently. Then on the other hand, I want to jump to stochasticity. So we talked about this. The quantum physics is really not opened the door for unpredictability, right? Basically, everything is in quantum physics is evolution of probabilities. It's not certainties. It's really a very sharp break from what 300 years of determinism in science, going back to Descartes and the French philosophers and so on. Now we know that that was an illusion, just an illusion. All right, so stochasticity. And then quantum information. So again, some mysteries of how we, and that's maybe something I observe myself quite often. I don't know if you have the same feeling. Sometimes you cross the street and in the corner of your eye you see somebody, maybe not the whole person, maybe just an arm or some part of the body or somebody moving in a special way, and you recognize right away, that's my friend. Although you don't see the face, and then you turn around and you say, yeah, that's him or her. Have you experienced anything like this? Quite often, right? So this is beyond, sort of standard, I would say, AI type of pattern recognition. This is almost imperceptible. Another thing is when you feel that somebody's looking at you from behind, right? You have the sense of somebody staring at you. Yeah, somebody's staring at me quite often. So how does it happen? Maybe this is another example of quantum entanglement, even between strangers in the latter case. In the first, yeah, friend or family member you can recognize from far away, and just maybe even without seeing the figure, but just movement and something like that. All right. We haven't talked about, well, I did actually last time. I talked about quantum statistics, so I'll just skip that. But that's another interesting thing which never before quantum physics was even contemplated, namely different types of organization of many objects. And so fermions, as you remember, Fermi-Dirac particles, they don't like to have anybody in the same state. They are very selfish. And then you have bosons, Bose-Einstein particles, which are the opposite. And electromagnetic waves, photons, are bosons. And phonons, photons, but electrons are fermions. So this is a new way of thinking about, away from actually Boltzmann, who also contributed tremendously to physics, but not to quantum. And according to Boltzmann, all things were equal. There were no different types of particles. All right, and then finally, on that list, and some of it is what quantum biology borrowed and used to define itself, is tunneling. Quantum tunneling, that's another bizarre phenomenon, but absolutely confirmed by a multitude of experiments. So tunneling is when you have a quantum particle which, if it was a classical, let's say, ping pong ball or a tennis ball, would not be able to go over a barrier. Let's say, I don't know if anybody plays tennis here. Do you play tennis? Nobody? So sometimes if you have no partner, you practice tennis by hitting against the wall, right? You hit and hit it. And sometimes the ball goes over the wall, but it has to have enough energy and has to have a special angle to do it. Imagine now that you play quantum tennis against the wall, and the particle penetrates through the wall, goes on the other side through the brick wall, even if it's at this height below the boundary. That's quantum tunneling. And quantum tunneling is manifested in all of the semiconductor technology, by the way. This is the basis of semiconductors, how the electrons tunnel in diodes and transistors and so on. And it's also present in enzymatic catalysis. So without quantum tunneling, some of the reactions would not be possible. One of which, by the way, is very important, namely in the electron transfer chain in mitochondria. Anybody here familiar with mitochondria? You have the inner mitochondrial wall, you have the complex 1, 2, 3, 4, 5, and then you have electrons hopping. This is a quantum process. Yes?
I heard in elementary school, my friend told me this, I don't know if it's true, I want to ask you, if you keep hitting your hand against the wall, for a million years, eventually your hand will go through the wall. Is that true?
Keep trying. Quantum hand. Well, maybe that's a new thing to explore. Well, of course you can break the wall, but in a quantum sense, I have some doubts. Let's put it this way. No, the electrons, yes. Okay, so I said a little bit about a few things about phonons. So remember, photons, light, phonons, vibrations, quantized, both quantum particles. So quantum theory of solids is where I mentioned, maybe I did, Einstein actually made a mistake, and there was, I don't want to criticize the guy because he did enough, but it's a fact. So these were the observations at the beginning of 20th century, that at low temperature, a specific heat of solids was proportional to the cubic power of absolute temperature. At high temperatures, it was constant, and R is the gas constant, you may remember from chemistry. So that was difficult to explain, and Einstein tried and didn't succeed. He got this result, which was exponential. Complicated, but incorrect. And that, by the way, that result applies to gases, not to solids. And this is an experiment for solid systems. Okay, so Peter Debye solved the problem, and I'm saying it because you'll see in a moment how you can almost completely transfer the mathematical model for this to the case of biological systems. So the Bayes model was that that you have these waves. I started by talking about waves, right, in solids. And instead of independent oscillations of atoms and molecules, each one moving around independently, which Einstein assumed and got the wrong result, the Bayes said, no, when you deliver heat to a solid, a crystal, you create these waves, like ocean waves, you know. These molecules move around together because and why is that? Why is that? Well, they interact strongly. They stay in the solid because of the strong coupling between them, right? You have the bonds. The bonds keep them together, and when you disturb them in their positions, then they hit or they transfers this energy to the neighbor, and the neighbor to the next one, the next one, the next one. It's a bit like, let's say, imagining. I don't know if you've been to a soccer game, you know, the Mexican wave, you know this? Soccer games, even hockey games too, they have the Mexican waves. It's a bit like that. So everybody's attached to a seat, but they have a little bit of freedom to move around, and they go up, you know, and then they go down. And so that's kind of how the Bayes envisaged energy transfer, and that costs energy, right? Mexican wave, you have to get up and down periodically. So that's the explanation. And he got the correct results. I will not go into the details, but we'll come back in a few minutes. Why keep this in mind, why this is important in biology. Synchronization again. So what happens to the solid? Synchronized oscillations. What happens to the human body? Efficient coordination. All right, so this is kind of a grand scheme of things in terms of dimensions, and so we're going now into why quantum physics is relevant to biology. You know, so all of this is well known to be within the realm of quantum physics, from quarks to neutrons, nuclei, atoms. Small molecules, if you go to biomolecules, large molecules, this is the gray zone. More recently, I think a group from the University of Vienna determined the wave function of a protein. One particular small protein, I think it was only about, I'm guessing 10 kilodaltons. I think it wasn't big, but they found the wave function, or measured the size of the wave function. So, all right, this is where the state of the art is, but since every bond is quantum, then we can say by definition all of it is. But in terms of descriptions in current practice, we then go on this scale of sizes from biomolecules to systems, let's say aggregates of macromolecules, right? And these are the numbers of atoms. 100,000, and a cell has 10 billion atoms on average. And the human body has 3.37 trillion cells. So you can count the number of atoms in the human body. It's a very large number. So you would say it's hopeless. You know, you really have no need for quantum because it's so many of these atoms, and we kind of stopped here at molecules which have 100 atoms, and this is quantum chemistry that does a very nice job of creating these molecular orbital representations. All right, and okay, then we have, so instead, that's quantum chemistry. Then there's mesoscale, where quantum biology is making inroads, and beyond that we have thermodynamics, mechanics, fluid dynamics, all the classical fields. And I'll skip that. This is just a table for anybody who's interested in numbers, as I am, as I told you that I had to remember a lot of these things. And this is two more slides to give you a perspective on scales, time scale and spatial scale. And that's why the challenge is so big. So this time scale of biological systems, sorry, this is the spatial, that's not time, time is next one. This is spatial scale. So what's the smallest thing that is of relevance to biology? We're not going to quarks, they are not relevant to biology, at least as far as we know. Well, the smallest is, I would say, the size of a hydrogen atom. Hydrogen is about 0.5 angstrom, so that's 10 to the minus 10 meters. Okay? Then we go to the nano world. And by the way, what happened to nanoscience, nanotechnology? Remember, it was all the rage about 10 years ago? Now we forgot about nanotechnology, it's all AI. the next fashion, I think 10 years from now will be something else. So nano is nanometers. That's the size of a protein. Proteins typically are several nanometers. And protein aggregates could be hundreds of nanometers to micrometers, 10 to the minus 6. The cells, bacteria, the smallest cells, bacteria are on the size of a micron, 10 to the minus 6 meters. Largest eukaryotic cells from tens of micrometers up to a meter, right? A neuron could be as long as that. Giant squid axon is 1 meter long. Okay, so, and then we can go to the size of the organism. Let's say the largest would be the elephant, is it? And whale, this kind of thing. We're talking about tens of meters. I don't remember how many meters an elephant might be. Let's say 10 meters. Maybe not 10, a few. Okay, it's 10 meters on that scale, the largest biological system. But you can also think about societies as living systems, right? And then you extend the scale to tens of thousands of kilometers. That's maybe something for the next lecture when we think about connectivity through the space of ideas and the mental space. All right, and here you have, that's very important. Here you have the corresponding scale of probes that give us information about these systems, right? So, because that's how we started with microscopy, you know, Van Leeuwenhoek in the Netherlands, the first microscope, and you can see cells. So, but now we can X-rays can give us information on the angstrom scale, and so X-ray crystallography. That's why we can resolve crystal structures because of X-rays. Then longer yet are ultraviolet, and then infrared and microwaves. All of this can be probing different aspects of living systems, and it is. And here you have pictorial representation of the corresponding sizes. Sizes of these wavelengths should be attuned to the size of the object that you investigate. So that's why X-ray crystallography can give you resolution up to typically 1 to 2, well, 1.5 I would say is pretty good, right? Angstroms. Not as hydrogen atom would not be resolved very well, but with heavier atoms and certainly positions of amino acids and so on will be. Okay. Now, so let's just look at the number of the orders of magnitude. So from, let's say, 10 to the minus 11 to 10 to the 1, 12 orders of magnitude in sizes. That's what biology, generally speaking, covers. And that's why it's so difficult to say, okay, we explain everything with one framework, conceptual framework, right? That's why we go from quantum to semi-classical to classical and so on. And this is even worse, because when you look at the time scales, and actually there's something cut off here, typical chemical bond vibrations, you know, vibrations and rotations and twists and so on, occur on the time scale of picoseconds, or even a fraction of a picosecond. It's very fast, very fast. And so when you talk about time scales, the flip side is frequencies, right? 1 over t is f, frequency. So when you say picosecond, that means what? 10 to the minus 12th. The inverse of it is 10 to the 12th. 10 to the 12th, inverse second is hertz. the unit of frequency is hertz. 10 to the 12th hertz is called terahertz. Terahertz, that's the probe for these kinds of chemical bond vibrations, especially water, by the way. And this is what you just got back from New Delhi. How many times have you gone through this?
A lot.
Terahertz. They use terahertz vibrations to scan, because the image is, the absorption of terahertz is mainly by water. What is not water is shown as dark. Okay. So then we have other scale. So we have, let's say, nanoseconds. It's not shown here, but nanoseconds, 10 to the minus 9, which inverse would be what 10 to the 9th hertz, which is what gigahertz, right? Gigahertz. It's 1,000 times lower frequency than terahertz. So nanoseconds correspond to conformational changes of proteins, and longer. That will be the beginning of changes of conformations of proteins. And then you have even slower, let's say, processes. Let me just mention 10 to the minus 3rd, which is millisecond. Inverse of it is kilohertz. Kilohertz. Milliseconds are typical processes where ion channels operate on. Ion channels on a millisecond time scale. And they would therefore could be coupled to kilohertz frequencies of electromagnetic waves or other waves. Another millisecond time scale is ATP production. ATPases, some enzymes. So in this particular case, important time scale for ATPases, FATPases in mitochondria. So you're all familiar, some of you, with mitochondria. They convert, well, starting with glucose, ending up with ATP. 1 ATP molecule per millisecond per ATPase, and there is, I think, around 50 to 100 ATPases per mitochondrion, and there is about 1,000 to 2,000 mitochondria per cell. Some more, some less, and that's the figure. And then you have even slower, so neurons, action of neurons, will maybe since we're talking about time scales, I'll ask you. So what do you think is the frequency, let's say brain waves. Do you know the frequencies, typical brain waves? What are they? Alpha, beta, gamma, delta. You're a scientist.
It can range, and like it has a big variation.
Yes.
So sometimes it's between like 2 to 10, and sometimes it's like 40 to 80, and
yeah,
even higher ranges such as
something like typically 10 to 40 hertz, right?
Yeah.
Gamma synchron is about 37, something like that, 30 something. So what does it mean? If it's from 10 to, let's say 100, 10 to 100, typically you look at the brain oscillation, E by EEG, that would be hertz. So you have to divide by this to get the time scale, right? So typical, so you 1 over. So it's 100 milliseconds, right? To maybe between 10 milliseconds and 100 milliseconds. Time scales on frequencies. And 1 millisecond per ion channel, and you know, so this is coupled to what action potentials going, you know, through the neurons and ions flowing in and out and all that stuff. So that's the time scale for the brain, and now it's still a challenge. So there's no good answer to that, but I'll ask the question: if this is the time scale for the brain, and yet we seem to agree that there is something mysterious in terms of entanglement, probably quantum, we need to have long-lived quantum states in the brain to explain the function. Maybe not all of it, but definitely some, right? So that's a challenge. Actually, it is not a joke. It's a serious scientific issue. How do you explain if the human brain operates at some stages and some actions, functions, maybe quantum? How do you explain the presence of long-lived quantum states? Because in the presence of noise, high temperature, you know, would standard quantum physics would tell you that it should decohere, that's the technical term, rapidly over picoseconds, but it's not. So it's an open question. Some of you may be interested in studying this in the future and getting closer to Stockholm if you solve some of these. Okay, so and last but not least is the energy scale, molecular energy scale of relevance to biology. So yeah, all of this has to do with processes, biological processes at the cell level, involve biochemical reactions, right? And here, bond making and bond breaking, the weakest bonds would be hydrogen bonds. And then the strongest covalent, you may know it, I don't want to belabor it. I just want to mention two things in this connection. Namely, in biology there is bottom of the energy scale in terms of relevance. If something is has less energy than kT, what is K? K is the Boltzmann constant, T is the temperature in absolute Kelvin. If something has less than that energy, it's irrelevant because it's basically would be washed out by thermal ice. kT is the free energy because we live at we live at 36.6 Celsius, and that's the energy we maintain our living state. If you lower the living state, the temperature of the body, and you correct me, Kim, but I would say hypothermia 35 would be probably close to lethal, right? For minutes or maybe several minutes, I don't know how long. So, and then 42 degrees Celsius would be fever, high fever that would result in a prolonged situation, death, right? So it's a very narrow range of temperatures, 35, let's say 34, maybe something like this, I don't know exactly, but around that, to 42. A few degrees. Amazingly, and again, I'm looking at you, correct me if I'm wrong, but I was told that all warm-bodied, warm-blooded biological organisms maintain exactly the same temperature as we do.
No, pigs are warmer. I know that.
Oh?
Yeah, not,
okay, so
maybe by degree.
Yeah. A degree. Okay, all right. I'm not going to split hairs over 1 degree Celsius.
Right.
But I was told by a neurophysiologist, he said to me that it's the same temperature. It's close. Okay, so it's very close.
Yeah, I think it is close, but it isn't
the same.
Identical.
Okay.
So when you see a pig, you know if you touched it, it would feel warm.
It cozy up to the pig. It's nice and warm.
Yeah.
Okay, so why am I saying all of this? Because this is actually this result, kT, at 36 is roughly 310, or 37, 310 Kelvin, corresponds to the energy of 0.6 kilocalories per mole. You know these units from biochemistry, right? kcal per mole. Okay, so that's the bottom. And the result comes from very sophisticated statistical mechanics. It tells you that every degree of freedom in a system of many particles, on average, has kT divided by 2 of energy, so 1/2 of kT of this product. That's for free, because the temperature equilibrates. Right? So our body, I think if we are healthy, again, I'm asking you to correct me if I'm wrong, but for under normal conditions, the entire body has the same temperature, right? More or less.
Except for the testicles, right?
Okay. Okay. We're not going to go in this direction.
You asked for it.
Okay. You see, in biology there's always an exception, darn it. In physics we have rules.
Yeah.
Okay, so yeah, by and large the same temperature, which means that the temperature is equilibrated, and that's the temperature for each degree of freedom. So every what's the degree of freedom? Every movement that is allowed would have this kind of amount of energy given for nothing. All right. So anything that is below, throw it out. It's meaningless. So that's the bottom of the energy. This is the top. I would say the molecular scale covalent bonds about 90 kilocalories per mole. And why is that? Just to maintain stability, right? Covalent bonds maintain stability of structures, and yeah. And the second thing, three things. That's the second. The third one, and probably most important, is this. This is the biological energy quantum. The energy of ATP hydrolysis, which is this universal currency, GTP to a lesser degree, but ATP is involved in every type of deliberate movement at the cell level, whether it be motor protein movement. Every step of a kinesin or dynein costs one ATP molecule of energy, and every step in the transcription and translation process. And what is this? Well, let's say that's kind of the average, 60 kilocalories per mole. So it's 100 times greater than free energy. So why is that? It's in order to prevent any mistakes from happening, because thermal energy is only 1% of it. It will not make a big difference, right? So these fluctuations, thermal fluctuations, will not disturb the program the cell wants to carry out, whether it be transport, whether it be motility or mitosis, etc., etc. So that's biological energy quantum. The first, okay, maybe I'll skip this. I just wanted in this diagram, I just wanted to say that we have compartmentalized science into fields. Each field, and we're getting into biology, but we started with, let's say, elementary particle physics. In this field, you have elementary particles and their interactions, and many of these interactions we ignore at the higher scale. If you go into condensed matter physics, you forget about quarks and in fact, even the nuclei, just electrons and protons make a difference, and then interaction, and that's what makes matter, condensed matter physics as a field. And then chemistry, ions, you ionize molecules and form bonds, and then biochemistry is, from chemistry you take a subset of molecules which are relevant to life. Nucleic acids, amino acids, sugar, phospholipids, and some biomolecules of other type, and you make biochemistry. Why is this important? Because by first of all, corralling elements or units into these groups, you create fields, and then the elements are given rules of interactions, and that's how you define a field of biochemistry. And cell biology will, of course, create larger structures and play on different cells, cell types, morphologies, and so on. Okay, so I'll skip that. This is kind of explaining to you at the moment why we cannot simply jump all the way to the human being and say everything is quantum, because our modeling, computational modeling and physical modeling, is restricted by our ability to actually perform such computations on different scales. And these are the time scales and length scales and different levels of modeling. So this is quantum mechanical models, picoseconds, nanometers, atomistic models, classical molecular dynamics type of things, nanoseconds, micrometers, and so on and so forth. So the objective is eventually to expand from the bottom to the top the ability of our simulations to capture the essence of systems, including biological, at a longer time scale and spatial extent. So this is one number that I often mention in my lectures. It's called the immense number. It's a technical term, immense number. So why is that? Because, again, I want to emphasize the limitations to our knowledge. And right away somebody will mention AI. I will preempt it. But why is this important? Because this number gives us a real barrier to knowledge. 10 to the power 110. What does it mean? This means the size, let's say, imagining that the entire universe, composed of all the elementary particles, was one big computer, and each atom, or yeah, let's say each atom, was encoding a bit of information and flipping these bits on the time scale of picoseconds, which is typical, as you remember, atomic scale of transitions. Subatomic, I should say. So if the entire universe was one big computer that operated for the entire existence of the universe, which is 14.5 billion or something like this, years, in terms of picoseconds, you may multiply these two numbers, you get 10 to the power 110. Which means that even inspecting every single object out of the set of 10 to the power 110 would take the age of the universe and the entire universe operating non-stop on a picosecond time scale. And that's the real boundary to our knowledge, a limitation. And you might say, well, but this is a ridiculously large number. What is so big? Well, many things. One of which would be our DNA has 10 billion base pairs. Not all of it, maybe 7% is non-coding, but we don't know. Maybe it is. As far as we know, it's not. But even if you only take 3% of 10 to the 9th, and there's 4 possible nucleic acids you can encode in each position, you'd have a vastly larger number than this to explore all the possible sequences of human DNA. Like, one example. Another would be, how many different proteins could you create out of the length of up to 500 amino acids, which is average, right? Average is 500 amino acids. Well, 20 possibilities in each position, 20 to the power 500 or more, at least. That's 20 to the power, this is 10 to the power 110. In other words, if you want to use brute force, computational brute force, and explore the space of all proteins from sequences of amino acids, forget about it. This will never happen, never ever. AI may give you some predictions or something like that based on the training set, right? Training set. So what would be the training set for AI currently? Well, we have about, what is it, 100, maybe 150,000 structures and protein structures in the protein data bank. So that would be the training set. Let's say 100,000, 10 to the 5th. 10 to the 5th, and we are talking about something like 20 to the power 500. It's ridiculous. So, I'm not dismissing AI, but I want to emphasize limitations to the human knowledge, even augmented by computers and so on. Okay, so now what is life? So this is, you remember this guy, Erwin Schrödinger, and of the Schrödinger equation fame. So this is not right. It's 1944. So was it 1933 he got Nobel Prize? Right, the year that Hitler came to power. I told you about last time. But 11 years. 11 years later, in exile, in Dublin, Ireland, he wrote this book, What is Life? And it's still valid today, and I highly recommend. You can probably get it for $10 on Amazon. It's a great book, a little book about how a genius physicist sees biology before DNA, before people knew that there was such a thing as DNA, and talks about information, biological information, thermodynamics, and asks the questions of this nature, including quantum coherence, and came up with this idea of an aperiodic crystal lattice, which is proteins. Okay, so yeah, we have to talk about evolution, Darwinian evolution, and the aspect of optimization is in the process of evolution. So think about this. Now we're talking about 2 billion years of life on Earth, roughly, right? 2.5 maybe, something like this. 2 billion years of life on Earth. And how many life forms started with, we don't know how, with this RNA soup, maybe something like this. We still don't know exactly how life started. But we know that there were gazillion, trillions of different life forms, and they, by the process of competition and adaptation, they refine their abilities to function. And that means, for example, it took us what? How long is human civilization, which means documented culture, I guess, written text and so on? Maybe 10,000 years maximum, less than that probably. Some paintings in Lascaux Cave in France, maybe 10,000 years. There was already some civilization because they were able to create images, right? Animals and so on. So I'd say 10,000 years. And in 10,000 years, we achieved, think about this, from primitive into, maybe not so primitive, but as far as technology goes, maybe primitive, humanoids or human beings who survived in caves. Now we reached the age of amazing technology, including quantum physics already 100 years ago. So 10,000 years for humans to understand quantum physics and employ it and create technological advances. Mother Nature had 2 billion years and trillions upon trillions of experiments, because every life form is an experiment, right? Everyone here is different. We're all experiments on evolution. So I think it would be incredibly arrogant to say that biology was so silly that they couldn't figure out quantum processes. And it wasn't, of course it wasn't. Okay, so that's Darwin meeting Schrödinger conceptually. All right, I talked about this. And I talked about, I'm going to skip it because I talked about all the criticism about why quantum would apply to biology, too complex, too warm, too big, too wet, and all of this, and every single objection to this can be countered by a solid scientific argument. And there are still unsolved problems in molecular and cellular biology, let alone human physiology, which all kind of boil down to these, first of all, collective behaviors, synchronized behaviors, integrated behaviors. Biology and life sciences in general are great in terms of dissecting the structures, understanding proteins, genes, protein-protein interactions. And now, since about 1999, so 26 years now, we are understanding interactions between them in the form of systems biology. It's a new field, systems biology, where integration of these interacting units, genes, proteins, and metabolic pathways is being modeled and understood. But global synchronization still is a mystery. You can use the sleight of hand and use some kind of domino effect, but if you carefully understand and analyze the time it would take, for example, even for things like signals reaching your receptors, eye receptors, let's say, I play tennis, not very well, but I do. And when you watch, professional tennis players like Djokovic, or in the past Federer, and their reaction times and perfect returns on somebody's serve at 200 kilometers an hour. You cannot explain this by molecular reactions taken, because even the signals going through the neuron from the optic nerve and then eventually responses to all the muscles involved in perfect return. I don't see how this can be explained by sequential processes. Kim?
So, Michael Levin and bioelectricity, I mean, he's
Yes.
talking about, you know, intelligence locally in different parts of the body. And that doesn't require synapses as far as we know. The knowledge is just there. And you can change it with an electrical signal
Yeah.
and so on.
Well, okay, it's a sleight of hand. Michael, I respect his work. He's a genius, maybe the greatest biologist of our time.
Yeah.
But it's not mathematically explained as far as I can say. I would like to see an explanation with velocities and, you know, and the time scale.
That's true, we don't have that yet. But I'm just saying that I think the two are somehow related.
Yeah.
The things we cannot explain yet, and his bioelectricity, when it's further
Yes,
explained,
yes.
probably relate to each other.
I get it, and I would say the following, that Michael stops at the electricity point, which is ion movement of ions.
Right.
I take one step further and I go into the electromagnetism. Electric currents of ions move at speeds of up to 10 meters per second.
Yeah.
Which is faster than chemical reactions, but still very slow. Electromagnetic waves propagate at 300,000 kilometers a second.
Yeah. Yeah. So
So-
that's fast enough.
Yes, and undeniably, electric currents create electromagnetic waves. Dipole oscillations create electromagnetic waves. So why do we stop at something which is so slow and so, of course it's documented, but we're going back to Galvani and Volta. This is 19th century physics.
Yeah.
I think there are better explanations if you include electromagnetism, photons, and now we are one step away from quantum. And entanglement, because you need to have the entire body in concert. And so yeah, you have all these parts trained independently, this muscle, that muscle, and the eye, and all that. But you don't see this, it's all integrated. And this is what I'm proposing, entanglement, a quantum entanglement of the entire human body.
Mhm.
Okay, so back to quantum biology, we have about 20 minutes left, so it's fine. So this may be updated now, I haven't checked, but Wikipedia definition of quantum biology focuses on these exotic aspects of quantum phenomena, not trivial like quantum chemistry, trivial. and so it talks about superposition. Remember the postulates of quantum mechanics, Solvay conference in Brussels? Superposition. In other words, quantum systems may exist in several states at the same time. Which, again, in the context of the human mind, I completely get it. I'm already thinking right now about what will be for dinner tonight. I'm in 2 states at least. Okay? Then, non-locality. Well, that's kind of tough. But we talked about non-locality entanglement. Non-locality meaning that it's not, again, Michael Levin's argument, complete locality, regional locality. We talk about non-locality. Maybe even entanglement with the tennis ball. Yeah, entanglement, kind of similar. Non-locality is a consequence of entanglement. And then tunneling. Yeah, and then tunneling. Tunneling is undeniably the case with many catalytic reactions in biochemistry, so that's kind of part and parcel. Now, historically, many of these famous scientists called for the development of quantum biology or for the need of quantum ideas in biology, and most of them were not biologists, by the way. It's almost like tooting their own horn, right? Physicists saying, yeah, guys in biology, learn from us. So we have Niels Bohr, Werner Heisenberg, Erwin Schrödinger, John von Neumann, Karl von Weizsäcker, Elsasser, Weizkopf, Eugene Wigner, Freeman Dyson, who passed very recently. He was one of the greatest physicists of our time, and others. they say that quantum mechanics is essential for understanding life at the most fundamental level. So we are scratching the surface, I mean, myself included, when you, let's say, study microtubule polymerization from tubulin, or how cells react to some stimuli, we scratch the surface, because there's still the concept of how cells are functioning as an entity, entire entity. Okay, so this is a list of some inroads into quantum biology, going back actually almost a century. Remember when we talked about physics? Quantum physics had 6 key experiments at first, which couldn't be explained very well, and that triggered the entire evolution eventually. I think in biology we're kind of at this stage. Quantum metabolism. I'll say a few words in a minute too. So biophotons, this has been a field kind of fraught with controversy. Biophotons are very low level emission of electromagnetic waves by living systems, above the black body radiation, so, you know, as a result of the function of being alive. This goes back to Alexander Gurevich in the Soviet Union about a century ago, and it's been poo-pooed, but it's now solidly established. In fact, I can tell you that something between 10 and 100 photons emitted per centimeter squared. I visited a lab in Prague in biophotonics where they measure these things. You put your hand in, they count the number of photons in complete darkness, and so on. So biophotons are an example of the emission of quanta from living systems. And the other thing is absorption of quanta, of electromagnetic energy, and the utilization of it, which is photosynthesis. And Barbara is studying how this phenomenon also plays a role in living cells, how living cells respond to different frequencies of electromagnetic radiation. So photosynthesis, I mentioned, light harvesting materials. This is a bit more, I don't want to go there. It's solitons, which are nonlinear waves, which losslessly propagate energy across scales. And that was proposed by another physicist, Alexander Davidov, in the 70s as a solution to the energy problem in biological systems. How energy is efficiently distributed, moved around, and so on. He proposes, and I'm going to stop here. It involves actually a coupling of electronic excitations, quantum excitations with phonons, and so on. If anybody's interested, I can give you literature. All of this stuff about cognition, whatever we're going to call it, consciousness, information processing, that'll be the last lecture, so I'm going to skip that today. And then there was another one, Herbert Froelich, who missed the Nobel Prize, should have been given, because he developed the foundations for what then became the so-called BCS theory of superconductivity. Bardeen, Cooper, Schrieffer, 3 Americans, your countrymen, got the Nobel Prize. Herbert Froelich didn't, but he laid the foundation, at least mathematically, to this. And then in the last decade or two of his life, he focused on biological coherence, that topic that I emphasize today a lot. And he came up with this theory of biological coherence, which is due to dipole-dipole coupling across membranes and cell-cell interactions electromagnetically, again, by electromagnetic means, not electrical, electromagnetic, because that would be instantaneous pretty much, almost instantaneous on that scale. And then there's some other Italians. Barbara is looking at me. I don't want to poo-poo the Italians. Giuseppe Vitello and Pepperata and Del Giudice emphasize the role of water in this quantum world of biology. Okay, so is quantum mechanics relevant to biology? gradually, more and more, I would say. And there are some examples which became famous actually in the recent decades. Photosynthesis, there were a flurry of science and nature papers, actually very famous papers around this time, demonstrating through experiments and computer simulations that plants, some bacteria and some algae, use quantum processes to harvest electromagnetic energy and transduce that for useful purposes such as glucose formation in plants. So, and the second incredibly intriguing and I think well explained is magnetoreception, magnetonavigation, in migratory birds. That was for the longest time, of course we know about this, right? For thousands of years, we noticed that birds fly from here. We haven't known this for thousands of years here, but in Europe we fly from here to California, let's say, South Arizona and Mexico. In the winter they'll be coming back. Canada geese, hopefully soon. And in Europe, the European robin and other birds go to northern Africa and Calabria too, Sicily, because
I think so.
they stay in, or just, I don't know if they stay in southern Italy.
It's like the pathways that way.
Oh, they fly through,
Yeah.
and they in the winters spend their winters in Africa, right? Northern Africa.
Yeah.
Okay, whatever. But they know exactly. Even the young ones that have never been there, they know exactly where to go. So the question was, how do they navigate? And apparently they navigate by the detection of magnetic fields and differences in the magnetic field. If they stray from the path, their brains recognize it. So this is copied from one of these papers. It's an amazing quantum process. Actually, there are molecules in the retina of the eye of the bird. So all of this is through photons coming in and triggering an electron state transition from singlet to triplet, and then the two states which are created by this excitation have different energy with respect to the magnetic field. So one is aligned. Remember, somebody asked me about the Zeeman effect, when spin, okay, now coming back, spin and magnetic field. So one state has a lower energy, one has a higher energy, and the higher energy electron comes down to the lower energy state, and that triggers a flow of charge, and that is then transduced into mechanical action. Well, what happens next maybe is not so well known, but basically it results in the change of direction of the flight of that barrel. It starts with photons triggering electronic transition state, and then magnetic field interacts with this high energy state, and then collapses it on the low energy state. And that, anyway, that's how it works. You can read about this beautiful, representation and use of quantum mechanics. Okay, now light detection by the human eye is another example that has been known for quite some time through experiments done by actually I think psychologists, and in complete darkness, and then they were shining a single ray of light, and apparently the human eye, which is not the best, I think, optical instrument. I mean, eagles and some birds have even more refined vision perception, but we can detect several quanta of electromagnetic waves in the visible range, photons, maybe not single, but 2 or 3, which is already a quantum effect. Then there is the sense of smell, olfaction. So for the longest time it was believed this is a typical receptor ligand fit, and that was dismissed because organic aromatic molecules don't really have some kind of unified shape. So that wouldn't work. The shape is not what is being recognized by the receptor. It's actually vibrational frequency. So it's a resonant quantum interactions, and that was explained by Luca Turin. He worked for a cosmetic company for 20 years or so, and he studied smell for commercial purposes. But as a result, found out that this our olfaction, and of course dogs and other animals have incredibly more sensitive sense of smell. A single molecule is recognized, right? That's why they use the sniffer dogs at the airport. Did you bring anything?
I was clean.
Okay. All right. So we have that. We have migratory birds, magnetosensing, photosynthesis, and then we have one more aspect which I kind of hinted at, which is mitochondrial metabolism, which is a quantum process because of the electron transfer, but it's more than that. So that's the vibrational theory of olfaction versus shape. Shape matching is not does not explain this. Okay, and maybe I'll skip this. There's something about there are some theories. I mean, this is not documented or verified experimentally, that genetic mutations may occur as a result of quantum tunneling. And John Joe McFadden wrote even a book about this and claims that our and maybe this actually makes sense in terms of even evolutionary theory, that there is no the mutations that, let's say, improve our adaptability to conditions, environmental conditions, and so on. It would be too inefficient if it was by trial and error and random mutations in our genes. He claims that it's actually the search, this quantum search for the best mutation would explain it, because we don't have to have so many futile attempts to arrive at the, so you try this, doesn't work, 99.99% of the efforts would be wasted. But by quantum search, you can speed the process up tremendously. So this was Froelich. I'll skip this and skip that. There's a lot of just long-range coherence in living systems was explained by this dipole-generated electromagnetic recognition. Okay, this is interesting. I highly encourage you to check out this website. You Google intelligence, cell intelligence, and you'll see like maybe 200 pages of work by Günther Albrecht Bühler from Northwestern University in Chicago, who studied cell migration as a result of the presence of other cells, and created situations where there is no physical contact between them, or chemical signaling. They are not touching, they are not sending, at least these molecules are not received by the partner cell, but they recognize the presence of other cells and they move towards each other. So he explained this by electromagnetic wave resonant interaction through, so one cell sends electromagnetic wave like an antenna sending a signal, and the other has receptors which recognize, in fact, these receptors for electromagnetic waves, according to Günther Albrecht Bühler, are centrioles. So also composed of microtubules, and these centrioles can recognize the direction of the light that is arriving at their position to within 3-5 degrees, in both in plane and out of plane. So they know exactly where the source of light is, the other cell, and that's how he explains at least this process. And then maybe I'll finish today with this kind of intriguing thing. So a lot of quirky stuff. today, but that's biology for you. We had 2 solid lectures on physics. Now it's fun with biology. The bystander effect. I don't know if you're familiar with the bystander effect. Yeah. So this has been studied quite a bit. There's a lot of literature on the bystander effect, and it's a lot of unexplained aspects, which could be explained by quantum entanglement again, because that means that cells, which are, let's say, for example, hit with ionizing radiation and damaged, signal to other cells in the neighborhood, and these that were not damaged, they also react accordingly. In some cases, the same applies, let's say, even to drug interactions. They somehow communicate to the neighborhood, and they share some enzymes and things of this nature. So stress-induced bystander effect, yeah, including radiation and chemotherapy damage. This person, she's a professor of radiation biophysics, I believe, at McMaster University in Hamilton. Actually, a distinguished scientist, a Canada Research Chair recipient, Carmen Madursil. She has studied these effects also in organisms, not just cells, but also organisms. And I, again, encourage you to look it up, because she reports some amazing effects, including experiments with schools of fish in 2 tanks. One school was stressed out, I don't remember, radiation, let's say, and then they were measured in terms of heat shock protein and some other expression, proteins being overexpressed. The other school, which was not, but in the neighborhood, expressed the same way. In other words, there was some kind of distance communication between these 2 groups of fish. And even more bizarrely, she reported and published experimental data on reaction to these stressors, radiation and other types, before the effect took place, before these fish were, they already perceived that for some reason. Okay, I'm just reporting this to you, which would be undermining causality. You know, causality is another of these long-held principles in our scientific thinking, right? Cause and effect. You have to have cause, then effect occurs. And believe it or not, but in relativistic quantum mechanics, there exist solutions of the so-called Dirac equation, which is like the Schrödinger equation, but with relativistic effects, where you have solutions propagating forward and backward in time. So if you have experiments which show you that there is effect before the cause occurs, that would be like propagation back in time. That sounds completely insane, right? Propagation back in time. But in recent literature in physics, you can find that there were experiments showing that there's exactly, on a very short time scale though, that has been claimed, let's put it this way. So quantum mechanics is able to explain a lot of strange things. I will stop at this, and the last lecture will, as I promised, discuss the human mind, neuroscience, and how all of this comes together. Thank you.
Thank you.
Yeah. So, any questions?
No, not today.
So we
Too
have
much.
A lot of questions next time. Okay.
Yep.